Okay. Well, good morning, everybody. And I guess if you can't see my screen, if somebody would let me know, that'd be great. I think I'm sharing the correct screen there for you. Appreciate you jumping on the call here this morning. So last week we talked about the basics of herbicides. Let me do one other thing. So we talked about the basics of herbicides as far as activity and things like that. Today and next week, we're going to jump into some of the more specific details around mode of action and site of action. We'll talk through many of the common herbicide groups, and we're just going to go in numerical order, the common ones that are used in production agriculture. And so today we're going to talk through herbicide groups 1, 2, 3, 4, and then the photosystem inhibitors. So 5, and 6, and 7, I guess. So for those of you that are social media buffs out there, some of you probably follow Bob Hartzler. So even though he's retired, technically, I guess, he posted, I think it was yesterday. I just happened to pull up Twitter and take a quick look at what the feed was, and he'd made a comment that one of his pet peeves was people mixing up mode of action, site of action, that type of thing. And I thought that was interesting because that's where I plan to start today before we jump into herbicide group 1. Okay, so as far as herbicide mode of action and site of action, there's a little bit of a difference there. So mode of action in general, it refers to kind of the whole process of what's happening when you apply a specific herbicide to the plant, the whole process from start to finish of how it's going to kill that plant. You can see a little more in-depth description there on the slide. Site of action, however, is a lot more specific. A lot of these herbicides are going to shut down a specific process that's going on within the plant. A lot of the processes that are going on within a plant are triggered by different enzymes to start those processes. And so with the site of action, that's talking about the specific site on, for example, an enzyme that the herbicide would bind to stop the function of that enzyme. And so there's a little bit of a difference there between mode of action, site of action. One thing that might help to explain a little bit better is this chart. So hopefully you have a copy of this Take Action Herbicide Classification chart. I've got a huge poster of it on my wall in my office. This information's in the crop protection guide in the technical information section that we talked about as well. It's structured a little bit differently than this chart, but this is a pretty handy chart to have on hand. I've zoomed in on a couple pieces here just to kind of highlight the difference between mode of action and site of action. In a second here, we'll start with group 1. Group 1 really isn't a good one to point out the difference between mode of action and site of action because there's one site of action within that mode. So the mode would be lipid synthesis inhibitor, and the site of action is this ACCase enzyme is where it's binding to. But one that would probably help to explain the difference between mode of action, site of action a little bit better would be the photosystem inhibitors. Okay, so these are the things like the triazines, atrazine, Basagran falls under this, metribuzin or Sencor products fall under this as well. But I guess the thing to point out is there's different binding sites within photosystem inhibitors. Okay, so there's 5, 6, and 7. And so this would be a good example of, we've got this mode of action, which it's inhibiting the photosynthesis within the plant, but there's 3 different binding sites based on which herbicide you're applying. Okay, so that's kind of the key difference there between mode of action, site of action. Mode of action is the general process, and then site of action is the specific binding site that's being stopped within that plant. Okay. Okay, so with that, we'll jump into Group 1, ACCase inhibitors, or so mode of action again on this is lipid synthesis inhibitors. A lot of times they'll be referred to as growing point disintegrators. With this specific group, you see there, so it's ACCase inhibitors, so it's actually acetyl-CoA synthase is the long name of that enzyme that's being stopped. With this, a lot of times we refer to it as our volunteer corn killer, or they're basically a grass killer. There isn't the same binding site within broadleaf plants, and so these only have activity on grasses. Some of the common products are listed, such as fusilade or clethodim. So Select Max, Section 3, those would be some of the clethodim products that fall under this particular herbicide group. The example of activity are shown in those pictures there. So after we apply a Group 1 ACCase inhibitor product, it's translocated with the sugars to the growing point of the plant, and so it's moving through the phloem. And eventually it'll arrive at this growing point. And so after you apply it, the corn might look green for a period of time, but if you go out there several days after application, you pull the whorl out of those plants, it should start to look dead like this. Okay, so it'll start, instead of being that nice yellow color, it'll start to look brown and mushy, and that's an indication that the herbicide is translocated to the growing point, and it's actually killing that plant. A lot of times the base part of the plant might stay green for a long period of time, but in general, the growing point has been killed. Okay. So with this one, we already kind of talked about site of action. And in general, what it's doing is it's shutting down that first enzyme in the synthesis of fatty acids. It's pretty much the building block of cell membranes that are required for plant growth. That's what we're stopping. I mentioned it's translocated through the phloem. Usually there's rapid absorption through the foliage. And it's translocated to the growing point, okay? And like I said, it's only active on grass plants. So a couple pictures from this last season. So we've got corn plant here. And, you know, we might look at this and think, might ask the question, what's wrong with it? And initially you might look at that and say, well, we've got a ton of Western corn rootworm beetles on this plant, which is correct. That is a problem. But if we zoomed out, the bigger problem is this is a volunteer corn plant, same plant in the middle of a soybean field. And so that's the bigger issue with this particular example, is that we didn't get good control of this volunteer corn plant, and now we've created a plant where the corn rootworm beetles are attracted to, and they're feeding there, going to lay their eggs. And so that creates an issue for controlling corn rootworm in the following season. Instead of being able to break that cycle by rotating to soybeans, now we've got volunteer corn plants out there, and they provided a alternate host or a host out in that soybean field, where we don't necessarily break the cycle of corn rootworm out in that field. And then the next year, when we rotate back to corn, we have increased corn rootworm pressure. Okay, so I bring this up because this year this was definitely a concern. There was a lot more volunteer corn out in soybean fields. So we'll talk about that for just a couple seconds here, where it's part of this particular herbicide group. So a lot of you have probably seen this slide on past trainings that we've done over the last year or two. This is from our volunteer corn demo that we've had out in the answer plots for the last couple of years, where we're highlighting a couple key points as far as controlling volunteer corn plants. And so to kind of orient you to what we did, In the top left picture there, that's section 3, so a 3-pound clethodim product, a group 1 ACCase inhibitor. We've applied it at 3 ounces per acre. And I will point out these pictures, we did take them about 2 weeks after application. And so we did use the correct labeled rate for clethodim, or for section 3 in this particular situation. But that is one thing to point out, you know, before we go and spray a soybean field post, and we've got a volunteer corn killer in there, we want to double check what the size of the corn plants are out there and make sure we're using the correct labeled rate for that volunteer corn killer. Okay, so 3 ounces per acre, but you can see we didn't do a good job of killing those plants. And so on the right here, what we've done is we've added in high surfactant crop oil, Superb HC. This is 2 quarts per 100, 9.6 ounces per acre at that 15 gallon per acre water rate. And you can see with that, with the added crop oil to it, we have much better uptake, much better translocation of the volunteer corn killer, and we've done a much better job of killing those plants. Okay, and so that's another key point, in addition to checking the labeled rate, that we're using the right labeled rate for the group 1 product, that we're including an oil in there with it as well. Okay. Next point here is a lot of these applications are going to be going out in a tank mix, and a lot of them will go out with a group 4 herbicide, which we'll talk about here a little bit later today. And so in this case, we've applied Ingenia in the bottom left at 12.8 fluid ounces per acre. Okay, we've gone with the same rate of section 3, same rate of Superb, but in this case, we haven't killed those. Okay, and so that's an issue.
I mean, I think I should be.
Antagonism that we see when we tank mix a volunteer corn killer with a group 4 product. Okay, ways that we overcome that. Main way is we bump up that rate of the volunteer corn killer. In this case, we've gone from 3 ounces to 4.5, as well as including that high surfactant crop oil. Okay, and in this case, we had very good control of those volunteer corn plants. Okay, so in a typical year, that's what we would see. This year, we did the same demo, and let's kind of walk through how it worked out. Okay, so we saw the same response up here at the top left. In general, we probably had, if we would have come back and taken a picture a couple weeks even after this, some of these corn plants had survived. So a few more of the corn plants survived even with the crop oil in there. Bottom left, we still saw that antagonism, and then bottom right, instead of it looking like this, and we totally nuked those plants, we had most of the corn plants made it through. There might have been a few there that died, but didn't see nearly as good control of the volunteer corn plants. And so we talked about this at different answer plot sessions. You know, what exactly happened? This was a pretty big challenge this year to control those volunteer corn plants. And what it came down to, if we think back to when those post applications were going out, it was really hot, it was really dry, so we didn't get very good uptake of the herbicide into those corn plants, and we didn't get good translocation of the herbicide to the site of action within the plant to kill it. Okay, when we get over about 90 degrees or so, those corn plants, and it's dry, those corn plants start to wrap up, right? And so that's a protective mechanism. Those plants aren't actively growing, and to get good weed control in general, not only with volunteer corn, we need the plants to be actively growing. And so that's why we didn't see good control of the volunteer corn. The plants weren't taking it up as well, and they weren't translocating it to the growing point of those plants. I threw this in here. Another issue with hot and dry conditions, those cuticles get thicker on the plants. And so this is another reason why we didn't see good control of volunteer corn in the hot, dry conditions. the cuticles, a lot of times we say the weeds harden off, the cuticles get a thicker waxy layer. So this is actually a picture from a microscope image, where on the left, this is a foxtail leaf that has adequate moisture, and you can kind of see the specks of wax on that leaf surface. But definitely when the plant gets into a stress condition where it's really dry, there's a lot more wax on the surface of that leaf. And it's the same thing is happening with volunteer corn plants in hot, dry conditions. Okay. Our recommendations for this next year would stay the same, where we'd say, hey, make sure you're applying the correct rate of volunteer corn killer. Make sure you have a high surfactant crop oil in with that mix. Some of them call for a methylated seed oil, so you can run that as well, or I should say a high surfactant methylated seed oil. And then hopefully the conditions are right, that we have good uptake, good translocation. And then if we are tank mixing with a group 4 product, make sure that we bump up the rate of our volunteer corn killer to better overcome that antagonism. Okay. Some key products that fall under this first group, we've already kind of mentioned them. Fluazepop, that would be Fusilade. Some of the other ones, Clethodim. A lot of the common products are here, Select Max, Section 3. And then we also have Post and Post Plus fall into that category as well. The group 1 products are broken down in different ways. For the most part, herbicides in general, there's a lot of microbial degradation that goes on. So you can see these have average, except for the DIMs, are a little bit slightly higher microbial degradation, average chemical degradation. Some herbicides are broken down in acid conditions. They go through acid hydrolysis. We'll talk about that more under the group 2s, and then the photosystem inhibitors as well. In general, these have relatively short persistence in the soil, about a half month. And then, yeah, so those are some of the properties related to the group 1 herbicides. These ratings are taken straight out of the crop protection guide. As far as control for volunteer corn, all of them are rated excellent, no matter if you're using the Fox or the DIMs. They all do a good job of controlling volunteer corn. As far as adjuvant recommendations, I'd mentioned before, you know, for the most part, our recommendation is going to be, hey, include 2 quarts per 100 of a high surfactant crop oil. In this case here with the Clethodim, you can see the labeled rate is a little bit, the recommendation is a little bit higher for Clethodim. Some of them might say you could use like if you looked at the Fusilade label, it would say you could use a NIS, so surfactant. In general, I do recommend using a crop oil just to do a little bit better job of controlling those volunteer corn plants. Okay. Particularly if we get into hot, dry conditions, we're going to want an oil in there to do a better job of burning through that cuticle and getting good uptake of the herbicide. So that's a little bit about group 1s. And I guess I will mention, if you have any questions, go ahead, throw them in the chat. We'll try to address them as we go here. Next group is group 2, so ALS inhibitors, the specific mode of action. These are amino acid synthesis inhibitors. Group 9s also fall under that mode of action. That would be glyphosate. We'll talk more in depth about glyphosate next week during next week's call. But today for group 2s, we'll focus on ALS inhibitors. Have some pictures here kind of showing the response of plants. If you're looking at this out in the field, trying to see activity from an ALS inhibitor, or if you suspect, you know, possible carryover injury from an ALS inhibitor, some of the things that you might look for. Top center here, bottle brush roots is a key indicator of ALS injury. And so that's what this is showing here. So you have your roots, but the lateral roots coming off those main roots are kind of short and stubby. They look like a a bottle brush. It's not very creatively named. Looks like a bottle brush from a soil lab. Okay, some of the other responses that you'll see, if we're talking broadleaf plants in general, if you flip over that leaf and look at the bottom of the leaf, a lot of times the veins will be this red or purplish color, and you can kind of see that showing up here on this leaf. Even on the corn plant here, you see a little bit of purpling of those leaves. Another thing that is a key indicator is you could have stunted the plants. So you shorten those internodes, and that's what we're showing here, top right. These plants were affected here. This is actually from an overlapped area on the edge of a field where it got a double shot. And so we did a pretty good job of stunting those plants. Top left, this is from one of our answer plots. The last few years, we've had a herbicide misapplication demo where we've tried to simulate carryover, in this case, simulating carryover of Classic into corn the next year. And so on the right, if we look closely, these plants are, at this point, they're about 6 inches shorter than the plants on the left. And this carried through all through the season. Even though the plants were nice and green, they were about 1 foot to 2 feet shorter than the plants that were unaffected by the ALS inhibitor. Okay, so you know, shorter plants, so stunted plants, bottle brush roots, and then the coloring of the veins is some of the symptomology that we look for. Okay, we'd already kind of mentioned it's inhibiting this specific enzyme, the ALS enzyme, and so what that's doing is it's inhibiting these the production of the branched chain amino acids that produce proteins. And so the plants aren't able to produce proteins, and eventually those plants die because they don't have the essential proteins. Okay. ALS inhibitors, they're absorbed through the roots and foliage, and then they can be translocated through both the phloem and the xylem. Okay. And depending on our soil pH, determines you know, how quickly they're broken down in the soil. And in general, we have a little bit better activity on broadleaves and grasses. Okay. After you apply an ALS inhibitor, you know, generally the growth of the plant stops within a short period of time, but then it's several days after that you'll start to see symptomology. Okay. So it's not an immediate effect as far as looking at those plants after they've been sprayed. Okay, some of the common products here are the trade names, Pursuit, and then Classic, and then in the right column you'll see some of the premixes that contain this specific herbicide group in it. Zidua Pro has Pursuit in it. That's a very common one on the market today. Some of the other ones here, so we've got Harmony, First Rate, Python, as well as some of the premixes that contain those products. If you flip over to some of the corn products, Accent Q, Resolve Q, as well as the premixes. If you're not familiar with that, Q indicates it has a safener to increase or I guess reduce activity on corn and make it safer to apply for corn. And then the final one here, Python, Option, Peak. These are some other chemicals that fall under this particular herbicide group. Okay, Python in particular, very common. If you're using Trizidual or Sure Start or Triple Flex, that's one of the 3 components, along with the stinger and the harness in that product. One thing I will mention here is just to watch out. We do have a stoplight chart related to this for many of the commonly used herbicides in corn and beans, as far as when it's okay to apply them, you know, pre and post, and when those cutoffs are for post application, so we don't injure the plants. I can send that stoplight chart out after the call with these slides. But with this here, just showing this is from an application of Accent that went on a little bit too late. When we hit that V5 to V7 time period is when the plants are setting the number of rows around on those ears. And in this particular case, the accent was applied a little bit too late. We ended up pinching those ears, okay? So that's something we're trying to avoid, okay? As far as how these are broken down, you can see low on the microbial degradation, high for chemical degradation. We'll talk about that here on the next slide as far as soil pH. Low surface loss from photodecomposition or volatilization. And then the persistence will depend on the product. Okay, so how does soil pH influence activity in the soil or persistence in the soil? Okay, so a lot of the herbicides, they become positively charged when we get into acid soil conditions. So the herbicides have a negative charge on, but then they grab a proton or an H+, a hydrogen ion, when we get into acid soil conditions, and then they're positively charged. And we know that the soil is negatively charged, so the soil grabs onto it a lot of times in the acid soil conditions and is able to tie up those herbicides. That's just kind of in general how that works. But with these group 2 ALS inhibitors, they react differently. These herbicides up here, Accent, Classic, and Canopy, these would fall under your SUs or sulfonylureas, which is a particular chemical family within this herbicide group. These ones on the bottom here, these would be more like your IMI. We're not going to pronounce that long name. IMI herbicides, which is a different chemical family within ALS inhibitors. But the SUs, these are broken down by acid hydrolysis. So you can see once we get above a 7.5 pH or so, that those don't break down as easily in the soil. And so we have more herbicide carryover of these particular products when we get into high soil pH conditions. On the flip side of that, with these IMI herbicides, the breakdown occurs by microbial activity, and this occurs more rapidly in high pH soils. Okay, so when we get into acid soil conditions, the breakdown is slow of those particular herbicides. Okay, and so there's some kind of differences there just within the ALS group of herbicides and how they persist in the soil, and it comes down to how they react with that soil pH. One thing I will mention here is because of how much ALS inhibitors have been used in prior years, you can see just how much or how many different resistant weeds we have to ALS inhibitors. It's a lot more than most of the other modes of action, okay? And so that's why as we go through the ratings here, you'll see a lot of them are rated as good to poor on a lot of these weeds. And it's because of overuse of that particular mode of action in years past, okay? So in general, with ALS inhibitors, performance is a little bit better with post applications compared to pre, and you can see that here. Most of these have a rating for, or aren't rated for pre-application, except when you get into some of these other ones like First Rate and Python. But even with those, you'll see it's fair to poor on a lot of these common weeds. For post applications in beans, a little bit more activity on some of these. Velvet leaf, for example, we see good to excellent control with a lot of these ALS inhibitors. Water hemp, not so much. Okay, if we switch over to corn with pre-applications of group 2s, same thing. Not much activity pre, more activity post, but still not the best ratings on some of these broadleaf weeds. As far as adjuvant recommendations for ALS inhibitors, we have a couple here, Accent, Resolve, nickel sulfuron, and rimsulfuron, where the labels call for NIS, so just your basic surfactant in there. whereas most of the other ones call for a high surfactant oil. So either a high surfactant crop oil or a high surfactant MSO. And then we have some other ones here as well. These ones are your common products for corn, and then these ones would be for soybeans. But still, adjuvant recommendations are high surfactant crop oil or high surfactant MSO. Okay, moving on to group 3s. So these would be your Dinitroaniline, so DNA inhibitors. These would be your root growth inhibitors. So mode of action is root growth inhibitor, and then the site of action is actually called Microtubule inhibitors. And we'll talk about that here in a couple slides, okay? As far as exactly what that's inhibiting to kill those plants off, okay? But some of the activity that we see out in fields, this is activity on corn. You can see the roots there, short stubby roots on soybeans. So a lack of development on those soybeans for the roots. And then you have swollen hypocotyls. In this one here on the left, this would be later on in the season after those plants have emerged. A lot of times you've weakened the stem if they've been injured with a group 3 herbicide on emergence. And so those plants could actually break off later in the season. Some of the common products under this particular group: Prowl H2O or Framework, and then Treflan or Trust is our private label version of that. Okay, I had mentioned before some of the site of action. On the next slide, we'll show you a diagram of that. So it's absorbed primarily by the roots. These are soil applied products. Generally, there's no post activity. These need to be incorporated because of photo decomposition, so breakdown in the sunlight. I know those have been, has been improved with new versions of some of these products, but still we're going to want to make sure we're incorporating these products fairly quickly after application, okay, just to reduce that photo degradation. Okay, so this is kind of diagramming the process of what the group 3 products are shutting off within the plants or within the cells. And so this is mitosis. If you're going back to your basic biology class, just showing cell division, where you start with one cell, and then at the end of the process, you have 2 cells that are pretty much exact copies of each other, right? But somewhere in this process, you have to double the amount of chromosomes, and then those eventually go to opposite sides, so you can eventually divide the cell. And so where DNA inhibitors are coming into that process are these microtubes, okay, or microtubules. And so those help with the development of these spindle fibers, which help to pull the chromosomes apart to opposite sides. So if we're shutting down this process, we can't continue to divide and make duplicate cells, okay? So the process of cell division is shut off with the application of DNA inhibitors. And so that's why you see those short stubby roots rather than a proliferation of roots, is the cell division process has been stopped. And so we end up with short and stubby roots, right? As far as breakdown of these, photo decomposition in general is high. I've mentioned before, we do want to make sure we get these incorporated 2-3 inches under that soil surface, so that we reduce this breakdown in the sunlight. Okay, persistence in the soil anywhere from 3 to 12 months. You can see some of the other ratings there as well. So this is showing it side by side, what the activity looks like on a corn plant if they've been impacted by an ALS inhibitor, so those group 2 products that we just talked about, or the microtubule inhibitors, so the DNA or group 3 products. So on the left here, we have the bottle brush roots, right, that we showed before. And then on the right, because we're impacting that or stopping that cell division process, we see short, stubby roots. Okay, so just showing that side by side. If you're suspecting herbicide injury by either one of these, that's kind of a comparison side by side to tell the difference in symptomology. Okay, two of the common products under this, pendimethalin or framework or prowl, those would be the trade names, or trifluralin, which is Treflan or Trust is our private label version of that. And then there is a premix of pendimethalin. Okay, ratings on this, you can see anywhere from poor to good for many of those broadleaf plants. And then in general, adjuvant recommendations, we don't recommend adjuvants for this specific product in the tank, just because these would be a soil applied herbicide, right? And so we're not trying to get foliar uptake. And so that's why we don't have an adjuvant recommendation with these specific products. I guess last thing I'll mention with the DNA products, I guess for maybe some of the newer folks or younger folks, is a lot of times we do refer to these as the yellows. They do stain equipment and pretty much everything else that they come into contact with. And so that's why I refer to them as the yellows. Okay, that's the group 3 products. A couple others here, group 4. These are the growth regulators. Some of the common products under this category, Stinger, 2,4-D, Dicamba, would all fall under group 4 growth regulators. The symptomology that we look for, you know, we have this epinasty is what it's called. It's a twisting of the stems. What we're doing with the growth regulators is we're impacting the auxin flow within the plant. And so we stop that, and so the plants end up twisting. They can grow calluses, like a lot of times you'll see after an application of a group 4 product, you can see the callus form down towards the base of that plant. And that's due to the impact of auxin flow within the plant, okay? One that I don't have a picture on here, I've got a picture on another slide, cupped soybeans. We're not necessarily going to dive deep into cupped beans today, but that is a symptomology of a growth regulator herbicide. On the corn side, this middle one here, this was from an application of status out in a field where it was applied a little bit too late. And so we laid those plants over. Also, if we had applied it with an organophosphate insecticide, that's broken down in through the same process within the plant. And so the plant can't break down both of those at the same time as quickly as it needs to. And so we end up bending those plants over, okay? So that's a little bit on group 4. I'd mentioned, you know, we're impacting this auxin production within the plant, or the flow of auxin within the plant. And so the plants end up growing themselves to death. You have this twisting or the epinasty that goes on. And a lot of times it looks like a slow death. It's generally 2 to 3 weeks on broadleafs before we fully killed off those plants. Some of the trade names that fall under this, Stinger or clopyralid, which is a component of that of Trizidual or Sure Start or Triple Flex. And so we see that's a component of those premixes. We also have Dicamba 2,4-Ds that fall under this particular group, as well as the premixes of those. So those are some of the commonly used ones in production ag. As far as 2,4-D, you know, there's different types of 2,4-D that we can use. There's esters, there's amines, there's acids. And so I threw this in here. This is a reference sheet. I'll send this out with the slides as well, because a lot of times we do get questions on, hey, here's the specific situation. Which 2,4-D product should I be using? Should I just be using an LB4 or LB6, or do I need to use an amine? And so based on, you know, if it's pre or post application, if we've got sensitive crops nearby, what else we're tank mixing in there, that kind of guides our decision. And so this is a decision tree to kind of walk through that process of identifying which specific 2,4-D we could be using. In general, the amines have better movement in the soil, and the esters, we see better uptake through foliar applications. And then rugged, or our acid version of 2,4-D, it kind of combines the best of both amine as well as ester. Okay. But like I said, this is just meant to be a reference sheet. I'll send this out after the call. One other reference sheet that I'll mention here is a lot of times we get to early on in the spring, and the question is, I want to apply a 2,4-D as part of a burn down prior to planting. Which product should I use? What rate should I apply? And how long, in particular, how long do I have to wait before I can go in and plant either corn or soybeans? And so there's small differences based on the product labels for what the pre-plant intervals should be. And so this is for corn. On this bottom half here, this next slide is for soybeans. So this is a one-page reference. I'll send this out after the call as well. Okay. A couple other things around symptomology. I did mention we see the cupping of the beans. This is an example here from the answer plots where we applied Extendamax, so Dicamba, to just Roundup beans, so they didn't have the Extend trait. And it's a pretty quick kill on those. Shortly after application, they start to look wilty and they eventually die. On corn, you can see fused brace roots, which is what this picture is showing here. And then on the bottom right, this is from a previous field call where they'd applied 2,4-D shortly before planting, and then it was cold, it was wet, and so they ended up having a little bit of uptake of the 2,4-D into a small percentage of the corn plants out in this field. But you can see we were out there probably V12, V14, and there's still a few plants that are tight onion leafed. Okay, so that's some of the symptomology out in the cornfields that you might see. One thing I did want to mention with where we're talking about the group 4 herbicides is just the importance of making sure we know what's out in the field before we go out with our post applications. Okay, and in particular, where we're on group 4 herbicides, we'll just talk about Enlist and Extend. So with Enlist here, it's these 2 rows that we planted through this demo, and this first 10 feet we sprayed with Extendamax. And so you can see if we spray Extendamax over the Enlist acre, we pretty well kill it off. And then on the flip side of that, where we have our Extend beans and we applied Enlist, or we applied 2,4-D, we pretty well kill the Extend beans. And so just a quick reminder to double check what we have planted out in the field before we go out with those post applications. So we don't end up in this situation. This is actually a little bit different situation where they had Extend Flex and some Liberty beans, and the farmer had thought he had all Extend Flex and then went out with Dicamba and pretty well killed off the Liberty beans there. Okay, as far as breakdown of the group 4 products, generally it's done through microbial degradation, some through chemical degradation as well. One thing I will mention here, we do have some specific group 4 herbicides for non. So picloram is the active ingredient in that. There's also products that have triclopyr, a few other actives as well. But those persist in the soil a lot longer. In general, we say it's about 12 months for the group 4 products at the longest. But Tordon can be up to 36 months. Okay, and so that's just something to be aware of. That's why a lot of times these are used on pastures or CRP ground where we're not going to be rotating back to soybeans very quickly. Okay, and I have also seen situations where Tordon's been applied to a pasture or a field where they've harvested the hay off of it, and then they fed that to the cattle, and then spread the manure on the field and wanted to plant soybeans there the next year. Well, that picloram molecule carries through the gut of the animal and into the manure, and then we end up killing off the soybeans the next year. Okay, so it does persist for quite a long time. So it's just something to be aware of on those specific products. Okay. Just showing the ratings here with Stinger, Dicamba, 2,4-D. In general, it's rated anywhere from poor to even some are rated as excellent on water hemp there. Okay. General adjuvant recommendations, and this is your like your Sterling Blue type product, your general or your Clarity, so kind of general Dicamba products, surfactant plus AMS, and then 2,4-D surfactant and AMS as well. Okay. So I will point out there is definitely a difference here between those products and when we get into our low volatility products like your Extendamax, your Ingenia, your Tavium. There's different adjuvant recommendations for the low volatility products that are used in season on soybeans. Okay. A huge difference is that we're not going to be putting AMS in with these specific products. Okay. AMS does increase the volatility of Dicamba, and so that's why we don't include it with either the Extendamax or the Ingenia applications. Okay, so general recommendations, Class Actinidian from Winfield, this is from Winfield United standpoint. So Class Actinidian, a non-AMS water conditioner, 2 to 3 quarts per 100. We include a volatility reduction adjuvant. That's new this year, right? And so our private labeled product is Volt Edge at 20 fluid ounces per acre. Or we also have Max NK that's been approved for Ingenia at 16 fluid ounces. It's been approved for Tavium at 64, and then it's in testing for approval as a VRA to be used with Extendamax. Okay, so we have some different options there. And then our final recommendation to go in with that is Ultralock, which is a combination of a DRT and a DRA. So like on target plus interlock, providing that drift reduction properties. Okay. That's kind of our top tier recommendation. I know some of you are probably have growers that are interested in an all-in-one solution. We do have some offerings in that space, some convenience products. I guess if you're interested in learning more about that, you can always talk to your local Winfield United rep about those offerings. But this is our top tier adjuvant recommendation for those Dicamba acres. Probably don't need to spend a whole lot of time on this, just showing what a VRA is doing. It's removing the protons, so the positively charged ions in that spray tank that would increase the volatility of the Dicamba molecule. And so we're reducing that to reduce the amount of volatility. For the Enlist acre, the adjuvant recommendation seems a little bit more straightforward. We don't have that restriction around AMS. And so our recommendation would be Class Act NG 5 to 10 quarts per 100, including a high surfactant crop oil, so 2 quarts per 100 of Superb, and then Interlock as our drift and deposition aid. And you might be using different products, but in general, it's an AMS water conditioner, a high surfactant crop oil, and a drift and deposition aid for the Enlist acre. Okay. And I guess one thing to mention is Corteva, they do talk about the drift aid that's included with the Enlist product in the jug. Most of the time, Enlist is going to be tank mixed with additional modes of action or additional herbicides or additional products in the tank. And in those specific cases, having an additional drift and deposition aid does help out. Okay. So the final one that we'll talk through here, Group 5 and Group 6, these are our photosystem inhibitors. There's different binding sites, whether it's a group 5 or a group 6. There's also group 7 as well. General symptomology here is shown on the soybean plants as well as the corn plant. These products are moved in a soil application. They're moved up and out through the xylem. So they move with the water flow through the plant. And so the initial symptomology that you'll see is around the margin of these leaves. You'll see that crinkling, that crispy burning, where it's been translocated to the edge of the leaves. And even on the corn plant here, you can see where it's been translocated to the edge of the leaf. So I'd mentioned it's moved with the xylem up through the plant. That's with a soil application. There's not much movement with a foliar application. In general, what these are doing is they're disrupting the photosynthesis within the plant. This is kind of showing the specific area of it. When the plant intercepts light, it excites electrons, and we've got this electron transport chain that eventually produces ATP and NADPH, which then go on to produce sugars within the plant. And so what we're doing is shutting down some of these proteins within that electron transport chain. And so what eventually happens is we shut that down, and then we end up with free radicals type of thing. You've got, and you end up with injury to the cell membranes, okay? And so that's how it kills off those cells, and eventually will kill off the plant, okay? Persistence in the soil, anywhere from 2 to 24 months. You can see the breakdown there. Similar to ALS inhibitors, there's differences based on soil pH. So in general, the triazine molecules, so they're more active at high soil pHs. And then when you get into low soil pH, they grab a hydrogen, and then they're tied up by either the organic matter or the clay in the soil. And so you have more carryover in those low pH soil conditions, okay? As far as activity, or not activity, as far as different herbicides that fall under this specific group, atrazine is probably the most common one, followed by metribuzin or dimetric or Sencor, and you can see some of the premixes listed there. As far as ratings of some of these products, good to excellent on water hemp and velvet leaf, so very good control of some of these key broadleaf weeds. As far as adjuvant recommendations for these products, it's different whether we're going out as a pre-application or as a post. For a pre-application, in general, we're going to put a high surfactant MSO in with those applications. For a post-application that has atrazine, we'll probably downshift to NIS, okay? So those are some of the basic adjuvant recommendations for group 5 and group 6. Okay, so covered a lot of ground here over the last hour. Appreciate you sticking on here and sticking with it. So this week we covered groups 1, 2, 3, 4, and then 5 and 6. So next week we'll continue on. I think we'll start with group 9, if I remember right, which is Roundup, and then move on from there. I did want to throw this in here just as a reference sheet. So I showed different pictures of herbicide symptomology. A lot of times we are called out to look at crazy stuff in fields and try to troubleshoot what's going on, or what application could have been made, or what carried over, or what drifted here and there. And we need to identify, based on the symptomology, what could it possibly be, which type of herbicide. And so I like this. This is actually from the University of Wisconsin. It's a 2-page sheet. I'll send this out after the call as well. There's similar information in the crop protection guide, but I like this. It's just a 2-page reference sheet, kind of a decision tree based on your symptomology. It gives you an indication of specific herbicide groups that could have impacted the growth of that plant, and if it was injured at emergence or injured post-emergence, So it's just a very basic decision tree to kind of walk through symptomology. Okay. So with that, I guess we're done here a couple minutes early, which isn't necessarily a bad thing. Any questions from the group? Don't see any in the chat. Okay, I guess we'll go ahead and stop the recording.